let's jump right in and start talking about DNA sequencing. And before we really get there, let's talk a little bit about some of the basics, and especially kind of what biology is about. And I think it's about answering the big questions in life. How did life begin? How do we have a concept where this is what we've looked at it for ages, and now we really start to see that it was more like this? One of the big mysteries is, how do organisms grow and develop? How does this really come from just two cells? How does consciousness hook into matter? How can this wet computer control this effector to produce something like this? One of the great mysteries that biology has really spoken to has been, how do we become who we are? Why are we like our parents? Why are we not like someone else or some other organism? And it's because of DNA. And being able to know the structure of DNA, and that's really because of these three people, Jim Watson, Francis Crick, and Rosalind Franklin, sometimes unfortunately overlooked, because when the Nobel Prize was awarded for this, she had died. But in the mid-20th century, they had gone through and found, through a lot of work and some good guesses, what the structure of DNA was, and that had implications for how it stored information, how it was able to replicate, and basically how the cell, in some ways, which stores all of this information, in fact, 3 billion bases worth of it, operates somewhat like a computer. So now that we've been kind of beavering away at what that means for 50 years, where have we gotten? And let's talk about some of the tools that we've got that kind of help us get through this. One of the things that's been around for a while now is the DNA microarray. It's really based on microelectronics, where we use some of the same technology to go ahead and deposit little strands of DNA in a matrix that go ahead and hook up with other complementary strands of DNA. It causes lights to light up in a detector, so we can say, hey, this person had this sequence, this microorganism had this series of bases. And in fact, I went ahead, I brought a brand new microarray from Affymetrix. So that was $2,500 to open that package, but that's okay, it was expired. If you guys want to take a look, wake you up a little bit in the morning, have something to pass around. And these have been now sort of a mainstay of biological research for going on 20 years in some form or another. Here's a company that I used to work for, did their own versions of this microarray, and one of the things they were known for was being able to get more things on there sort of faster and cheaper. I don't have one of those, but you guys indirectly are interacting with one of those, because this is the microarray that's used by 23andMe. Hey, Carlos, could I get you to start my timer? Timer for me, please, sir? Thank you. Or I'll talk to you guys for about 3 or 4 hours. So the other big tool that we're now starting to use is DNA sequencing, and there are several ways to do it, but the way I'm really familiar with is you take something that is the size and shape of a microscope slide, and it's got 8 lanes embedded in it. And you go ahead and you flow DNA through a little hole in the end of one of the lanes, off the other one. Some of that sticks, and you do a reaction, the polymerase chain reaction. And this is great. This is the exponential basis for everything that we do in biotechnology today. And what happens is you take one strand of DNA, and in a process that echoes what happens in cells, and by hijacking one of the enzymes that we use in cells to reproduce our DNA, we go ahead and heat and cool the DNA, and we make a copy of it. We make one copy the first cycle, 2 copies the next, 4, 8, 16, until you go through, and this will start to sound familiar to you guys after a while, about 30 cycles equals 1 billion copies of DNA. So then you've got enough to actually even see with the naked eye. You've got enough to work with. You've got enough to do detection or cloning. And here there's some clever tricks that are used to make these light up, and different bases light up in different colors. We use an optical scanner that is basically the twin of the scanner that's in the Hubble Space Telescope. But to peer at this one little place on one of these flow cells is what they're called. And these were actually used to run human genomes. I'm going to go ahead and pass these around. They're a little sharp. They're made of tempered glass. And somebody in the last class had dropped one, and so look out for a jagged edge. So by looking at billions of these points, we can go ahead and feed this all into a computer, and the upshot is we get DNA sequence. And people have now been doing this for a while, and we have one of these great Kurzweilian charts that show you, hey, here's how much raw sequence we've had, here are how many sequences we've had deposited in something called GenBank, which is a government resource, where they said, hey, if you're going to publish anything that's government funded, you do some kind of genetic sequencing, we want to have a copy of it in a public database. And anybody, you, me, high school kids, can get to this database and look at all these sequences. And notice a couple of things about this chart. One is it's an exponential chart, so that nice steady growth means that's exponential growth. You've got some doubling going on there. And notice also that it stops in about 2008. And what happened was a couple of things in 2008. The government agency that was doing this said, we're getting so much sequence, we really don't have a place to put it anymore. Kind of like Mom did with my comic books whenever I moved off to college. Don't bring anything else home, you know. In fact, every time you leave, you're going to have to take something with you. They ran out of space to put anything. They started talking about, what are we going to go ahead and do? We'll have a thing called the Short Read Archive. That's just been canceled. There's so much coming in. And part of that was because we started to see these next generation sequencing machines around 2008. So in 2008, they stopped taking submissions, basically, and that public figure for having several terabases of information has been stuck. This is what it has looked like as we've gone through and sequenced the human genome. The Human Genome Project originally ended, more or less, in 2001 after 13 years. And some people say $1 billion, some people say $300 million of funding. And parallel to that, Craig Venter has started a new kind of sequencing where he said, hey, if we go ahead and race these guys and we get there first commercially, we can wrap it up and patent it. And because he started later, and because he applied, you know, some business principles, they were able to finish in 3 years for $70 million. And that sort of thing has really told the story as we've gone through. James Watson's genome, one of the discoveries of the structure of DNA, they ran that in 2 months for $1 million with a company called 454. Illumina did something, and I want to talk to you a little bit about this, the Urubin Trio. So let me mention this now. In Christmas of 2008, we had several of these new sequencing machines come online. We said, let's see if we can figure out exactly what they can do. There was a mother, father, and child who had had samples in Africa, and that has been sort of this neglected ethnic area where we haven't looked at as many sequences, and it's got the most probably sequence diversity of any place in the world. So we said, let's go ahead and just run these things day and night. We'll have people come in and check them. We'll go ahead and figure out what we're doing as we get more stuff in. And what happened was that in about 6 long weeks and weekends, we came up with more DNA data from these 3 people than had been recorded by every scientist in the world working on this, in not just their data, but on any DNA data on any organism for the last 20 years. And that is what it looks like. We got a GenBank equivalent. And remember that because we'll talk about that a little bit more. So people ask, how long is it then until we get to a $1,000 genome, until we get to a point where these things are getting cheaper and cheaper, it doesn't cost much to run. And I really think the more interesting question is how long is it till we get to a $1 genome, and what happens when we get there? Here's one answer. Jun Wan is this chief scientist for the Beijing Genomics Institute, which has recently been renamed morphisms in DNA. But if you look at, sometimes you have more than one copy of a gene, or even more than one copy of a chromosome, or bits of chromosomes that break off and rearrange. These structural variations, these copy number variations, can really have a profound effect on how genes are expressed in your body. And it turns out there's a whole field called epigenetics about a code that rides above the DNA that turns genes on and off. The neat thing is sequencing can address all of these. You can use this DNA sequence with a few little tricks to see what the copy number variation is, to see which genes are expressed when and what kind of cells, to see these epigenetic changes. So I want to very quickly and shallowly hit on this just so you guys have an understanding. This is a current area of research and a lot of stuff going on. So epigenetics really just means above genetics. So it's a fancy way of saying there's another code that rides on top of this C, G, A, and T code. And we know one of the ways epigenetics works is sometimes the C, the cytosine in DNA, can be altered and methylated. And what that means, that lollipop sticking up is supposed to represent a methyl. So the protein that would come down and cover the DNA to recognize that piece or to promote the expression of a gene can't get to it. It's blocked. And so that can have sometimes profound effects. DNA methylation is why twins who are genetically identical actually are different. It's why sometimes diet can affect genes, and this can actually be passed on through different generations. So this is really interesting to me because I've got twins, and my twins sometimes are very different. One of the things that I enjoy is apparently one of them has the methylation state that makes him Dr. Evil. We're not sure. The other one is not at all evil. So, whenever you look at DNA methylation, you can see this as a switch that's on whenever the C is not methylated. Here's a region of the genetic code of the DNA that proteins can access and get to. Whenever it is methylated, it's off. And I just want to show you, this is a horribly complicated result, but it's from an experiment we did where we ran a full genome methylation scan of breast tissue, normal breast tissue, and breast tissue from a breast tumor. And we were able to see these really different methylation states. What happened here was this was upstream of a gene that ordinarily should be turned off. It's a growth promotion gene. You'd only have it whenever you were, very young, going through massive tissue growth. The upper part of this graph shows on the top and bottom strand of the DNA, there's high methylation states in several different places. Each one of those lines represents a different base where this is basically, this gene is blocked. But if you look in the cancerous gene, the cancer has found a way to eat away at this methylation where at least, in a high percentage of the genes, they're not methylated and this growth factor is turned on. And this is one of about 200 really clear examples of how we saw that methylation was being used to regulate the growth of these genes. I want to mention transcriptomics, which is a fancy way of saying the changes in gene expression. And what it is, this isn't just sort of you've got DNA, you've got 20,000 genes, everyone is on. Or some of them are on and some of them are off, and if it's either on or off. Sometimes you have a gene that's expressed in one tissue in a cell, you've got maybe one copy of that protein. In another cell, you've got 20,000 copies of that protein. And different cells have different genes on and off and at different levels. We can use sequencing to figure that out. This is something that's been done with microarrays, but we can now actually take all the RNA out of a cell that is expressing genes. So an RNA is the way that you have DNA sort of sitting in the read-only memory, the nucleus of the cell. RNA is what you go ahead and you make a protected copy of that protected genetic information, put that out into the hustle bustle of the cell where it's used to make a protein. You can control the expression with that by if you make 42 copies or 42,000 copies of that gene, and you use each one of those to make a protein. So we can go ahead and take that RNA, sequence it directly, and get an idea of a snapshot, the active state of the cell. This is how many of each of these genes are in two different kinds of tissue. Here we're showing brain tissue and a reference tissue. And it makes more sense whenever you put it on a graph and you can see, hey, in the upper part, the brain, some of these things are on or off, and they're on or off to a greater or lesser extent. In this average universal control, which is a mixture of tissues, you can see some things are almost always on. And this was an experiment I'll just mention we did with about 20 different bodily tissues and did this sort of human body atlas, really, where we were able to look and get an idea of what standard genetic expression is for each of these tissues and for a lot of different cell lines. We were also able, and I don't want to go into detail here, but see alternative splicing, see how different genes, instead of just having one form, you can actually turn bits of them on and off and express them in different forms. And we used to think that it worked a lot like that red figure on the right. And it really turns out that it's much more complicated and you get a lot of different forms. It looks more like the blue and the yellow figures. Those kind of structural changes too are one of the things that can be responsible for cancer, because you can have two bits of DNA that ordinarily would never come together, be spliced together by a cell that's in distress, and you end up with a gene under control of a promoter region that turns that gene on so you get explosive growth. And I just wanted to mention there's a thing that's relatively new in the last 10 years. Nobody even knew about it 10 years ago, and now there's been Nobel Prizes won and drugs developed based on these little bits of RNA that float through a cell. And it's just important because it turns out that these little bits of RNA that we thought were junk, we thought were just, what happens whenever you chop up RNA, are actually regulating all the genes in the cell. And again, we can sequence those, see what's going on, make drugs based on those, inject those into tissues and cells, and turn genes on and off. So the whole point of this is not to understand in any kind of detail epigenetics or gene expression or microRNAs, but to know that there's a lot of things going on, and that probably we're not going to get a single vanilla gene sequence in our lives. Babies are not going to be born, get a little punch on the foot, and then they hang a DVD around their neck saying, here is your DNA sequence. Although I think that will happen, we're also going to see this technology used for a lot of other things. And that's good that it's getting so cheap so fast, because all of those other things are going to require cheap, efficient, nearly constant uses of this to scan us and our environments. Here, I just wanted to make a real quick mention, and I'm going to skip over most of this. Sequencing is not the whole story, and sequence doesn't really tell you who you are. Keep in mind that, at least half of what you are is your upbringing, your environment. Different diseases we actually are able, before we had DNA sequencing, we knew from looking at family trees, things run in families or they don't. So the idea that your gene map is not your fate map. There are some things that surprisingly it does determine, and I think, as a society we're going to deal with more and more things like that, but not every little thing. Then I would go into this better, faster, cheaper. What are some of the things going on in technology that are going to make different technologies work better? What are the trends to watch for there? But let's skip that and talk about really quickly a couple of emerging trends. And I think this is kind of my big take-home message. Biology, like computers used to be, is now really under Moore's Law. And that makes sense whenever you think about Moore's Law. What is Moore's Law? It was this way to say that throughput increases every year and a half or 2 years because as feature size gets smaller, you have faster computers that can operate in less space, to some extent with less power, but with less lag time between pieces on a board communicating. Well, that same decrease in feature size really is driving this same change in biology, because as I can get more specks of light on those little flow slides, and as I can get a better camera that can distinguish more specks of light, I've got a multiplier effect with Moore's law, the electronics letting me do better biology. In fact, that multiplier effect is at this 5x, and it's looking sometimes like maybe it's going to 10x a year compared to the Moore's law. So the DNA throughput is improving on a yearly basis, not just better than Moore's law. It's not just beating Moore's law. It is really, really beating Moore's law with a stick. And so keep this in mind. And I don't just mean to be cute, although I do. This has, I think, a profound effect on the economics of this industry and what we're doing. And here, again, get used to looking at these exponential charts. Here you have the price falling exponentially, and you've got two different technologies. You've got your informatics technology in blue with this 2x decrease, nice steady decrease. You've got your sequencing technology falling at about 5x, it's in green. What happens is that even though the pie chart, which is not to scale, gets much smaller every year, the proportion of that pie chart that is due to using computers to store, analyze, visualize, do anything with this is getting bigger and bigger, more and more costly proportionally, and more of a bottleneck. And really, this last year, we passed that halfway point. So biology, in a very real sense, is not anymore a biologically constrained science or technology. It's an informatically constrained one. And the people who understand the informatics are going to be the people who drive it. Sequencing now, in some ways, is becoming a commodity business because we're racing to the bottom on our pricing. So you'd really probably rather, instead of trying to compete in that race and shave another cent, per million bases off of this price, you'd like to be the person who uses all this cheap sequence coming up. But don't think that that means that this is no longer a neat business to be in. The cheaper sequence means there's more and more market for it all the time, and it seems to be bottomless. And I just want to make a real quick point here. I don't know how much we're going to talk about nanotech in this week, but really look at the advances made in sequencing technology as a route to developing nanotechnology, by which I mean controlling molecules on the nanometer scale. What we've done is we've hijacked Mother Nature's way to do this by using some of these enzymes, and whenever that happens, we don't have to pay patent fees or license fees. We're going ahead and taking something we already know that works and improving upon that a little bit. So there's sort of this wet nanotechnology route to having molecular control. In some ways, like people who improve RAM for microelectronics, that's where all the action is, because you've got to make smaller and smaller feature size. Sequencing is that same way, I think, for nanotechnology. You've got to figure out how to do things with smaller and smaller molecules and better feature control on a small scale. So I hope Ralph Merkle will get a chance to talk to you guys about nanotechnology later on. We made the point, biology is now an informatics discipline, and it's now personal. And this is kind of my favorite part, so let's talk about this a little bit more. So we say biology is personal, and it's becoming more so. And is it really? Whenever we talk about the personal application of these technologies, we're assuming, there's a lot of pounding on tables, the revolution's here. We've got a 5x decrease in cost for the throughput. Is that really true? Well, it's apparently true if you've got $500,000 to spend on one of these machines and a PhD in some sort of molecular biology, and you're in a lab that's equipped to use this and has all the permits, which, is not really the innovation profile that we like to see in Silicon Valley. And the truth is, there are a lot of things about biotech that are really hard. And to give you an example, let me talk to you very quickly about my favorite project in my whole life, the thing, well, my second favorite that I've worked on. It was this. Mom and Dad got this for me when I was 8. And it was from Radio Shack, and it was my first computer. And it was a D-cell battery, some lights, and some wires. And if you look really closely on the box, and this is actually a photograph taken at the Computer History Museum, they've got one of these on display. It says in there you can use it to predict the weather, which, yeah, that didn't work out so well. But the point is, you have this moment when you get to build something and you understand how it works. When you do a computer program and you can do Hello World. And that is really hard to do with biology because it's complicated. Or is it? So here's a project where some bacteria were set up as light sensors and exposed to light so that they would change colors, and they can do Hello World on a gel plate about this big. There's about a megapixel per square inch in there. And this wasn't done by 3M, and it wasn't done by Pfizer, and it wasn't done by the Department of Defense. It was done by a bunch of students at UT Austin, one of the first iGEM teams. And you're going to hear some more about iGEM later this morning, so I won't go into a lot of detail, but iGEM is the Internationally Genetically Engineered Machines Competition. So there are now college and high school students who are doing these kind of projects and getting to Hello World really quickly. Biology and biotech is now not just an ivory tower science. There are people who are doing this all over for their own purposes. So tomorrow night you'll get to listen to David Rose, and David is this great speaker and this great proponent of crowdsourcing, offshoring, rapid prototyping, and the whole Web 2.0, 3.0. Think about those principles applied to biotechnology and medicine and see what you think. And let me give you a couple of examples of some of my favorite things that I'm seeing. So Garage Labs. Here's an actual garage lab in Silicon Valley, and it's not even a 2-car garage. This garage was something I had kind of stumbled across, had met some of the people who were working here about 3 or 4 years ago. It was better equipped than the company I was currently working at, which was like capitalized at $3.5 billion. They had a better lab from stuff that they had found in dumpsters and repaired, from stuff that had been thrown out because somebody had the latest and greatest thing and they didn't need the 2-year-old version of a lab robot or an incubator. And they were doing really good, serious work. You can see, and they've got a safety hood, they've got an incubator, they've got the biorobot there. This is a different configuration in the same space when they were doing tissue culture. These guys were working on a technology to take your innate immunity and use that to fight cancer. Because wouldn't you like to not take any drugs and just have your white blood cells go after and attack cancer? It turns out that that's what happens most of the time. What if we could go ahead and find the person who has the champion white blood cells and induce that same kind of fighting spirit in our own white blood cells so that this would happen? And so the guys who did this actually went on to make 2 different organizations. One was ImmunePath, which these guys went to animal testing of their product with less than $1 million in angel funding, and that's just unheard of. I mean, by like a factor of 20. And they've got really good results, and in fact, I'm hoping we might talk to one of these guys later this week. And they are now in the middle of their Series A and have done amazing work by, kind of using things that they found. The other person went on to found BioCurious. And BioCurious is a hackerspace for biotech. It's a community lab. And the idea is that you can experiment with friends. So, and we'll talk a little bit more about that in just a second. But as you have more and more tool makers getting involved and making cheap tools for these things and sharing the knowledge of how this is done, you're getting this multiplying effect As an example, almost every lab has some kind of centrifuge. And if you look at it, these are these fairly expensive precision pieces of equipment, but really all they do is spin things in a circle, right? Well, what if instead of buying one of these, you had one of these? This is, and you guys are going to look at some more of this later, it's a 3D printed piece of plastic where plastic has been deposited in a matrix, printed from a computer file, and you fit the test tubes in the little pieces there, and it's a rotor, and then you take the rotor and you put it on a Dremel. And it works just as well. And you can either buy one of these on Shapeways, or you can download the plans and put it in your neighborhood 3D printer and make a Dremel fuge. That costs $57 versus, even for a cheap used one, a good microcentrifuge is around $500 on eBay. And the shipping, don't even get me started on the shipping. So these are the kind of projects that are going on. Here is an actual commercial product that was this really innovative thing. The industry had made these real-time PCR machines, these copy machines for DNA that you have to get anything done. And they were charging tens and tens of thousands of dollars for it. These guys, Helixis, came up with some really great wrinkles in the technology, some shortcuts, and said, you know, we can do this for under $20,000. We can do this for around $14,000. And this was kind of a neat thing because instead of buying, you know, some $50,000 machine that goes in some central building and everybody from different labs, from research scientists has to come and use it, you can go ahead and have one that's maybe on a bench top in one lab with 3 or 4 or 5 people using it, or even an individual using it. And so they took advantage of this. They said, hey, we'll personalize it. We'll, you know, this is your iPod of the lab bench, and you can get skins for it. And it was really cute. And I got a chance to interact with the guy who was the president of this company, a great engineer, a great businessman, and he had gone through and done this good thing. And Illumina had acquired his company whenever we were on this shopping spree. And we talked about the competition, and I said, what do you think about, you know, these prices falling? He goes, well, we've got really great quality, but it's going to be hard to compete with us from the bottom. Nobody's going to be able to get a price like this. I said, really? Well, what would you think if somebody could do that for $600? He goes, I wouldn't believe it. So 2 guys, one of them had worked for eBay, the other one for Cisco, said they wanted to get out of this computer programming thing and break into biotech. Let's look at the tools of biotech, they said. Let's do something. Let's make a PCR machine and make an open PCR machine. We need about $5,000 to get started. We'll go to Kickstarter.org, where, you know, indie artists go to say, I need to do my next album, give me some money so I can do it, and you get a free copy. They said, we need to do a DNA copying machine. Give us a little money and we'll send you one. They raised the money, they went ahead and finalized their design, and they built one. And the neat thing is, you can go on the web now and buy your own DNA copying machine for $600. Or if you don't want to do that, you can pay less and get the parts and build your own DNA copying machine. Or if you don't want to do that, you're really cheap, you can go ahead and they very happily provide the plans right down to the circuit board designs, the assembly plans, and the patterns for cutting out the case with a laser cutter. And you can go ahead and not just cut out your own parts and build it, you can improve on this design because it's an open source design, re-upload it, and they will say, here is OpenPCR revision 2.13.1. That is the brave new world that we're in. So at BioCurious, I wanted to mention, we opened this lab space up, and it's in Sunnyvale. We actually built our own lab benches. We got this started this summer. It just did its grand opening a little over a month ago. And one of the things we kind of rushed up because we had a class of Singularity U graduate students in for the summer program, the 2.5-month program. And we wanted them to do some interesting things. So what they did, people who had never done this before came in. One of the former students from Singularity U ran the class. This is one of these guys who, you know, can figure out, oh, well, you need a Canon, we can probably build it out of these spare parts. He said, okay, we're going to do DNA sequencing, and this is what we're going to do. Show people who had never done it before how to extract and amplify their own DNA. We did that after we actually built our own PCR machine. Here's some students actually looking at their cells as they're getting it extracted. And we got it out to DNA sequence, and we did that in a couple of 3-hour sessions in less than a week. People who had never done it before. Here's another example of something at BioCurious. You see the little petri dish there with the lights on it? That's an E. coli protein that is glowing green because it's doing something no E. coli protein has ever done naturally. It's been genetically engineered to have a particular protein out of a jellyfish that glows green. And it's, kind of a simple genetic engineering stunt, but it's genetic engineering. It's genetic engineering 101. Here's the 8-year-old girl who did it. And she's actually showing these guys how to do it, which I love that. And the guy behind her is a scientist at one of the sequencing companies in town who had been helping out the class. She's like, oh, no, I've done this before at home. Let me show you guys. And so we are at a point where the tools are that pervasive and that easy to use. Why is it a big deal? Because this democratization of technology is letting the street find its own uses for the tools, and people are using these for amazing things. And every week something comes up and I'm just stunned by it. I truly believe, and this is my personal opinion, this is my raw opinion, but I think we're getting ready to go in this biotech-driven renaissance in medicine and manufacturing. And just the tools are there, the groundwork is there, people are starting to do their own things. It's amazing. But I know for sure that by doing rapid prototyping and having a community that is sharing these techniques and it's rapidly evolving, for me personally, it's totally changed the way I work. I'm like the gray eminence at BioCurious. I'm one of these old guys. There's all these 20-somethings running around getting things done so much faster than I ever have in my life, and it's changed how I do my personal projects and my professional projects. It's a new way of working. So, direct-to-consumer genomics, I want to mention that as another personalization trend. I want to put in a quick plug. Genomera is the company that I am working for and with, and we're doing some interesting things there. You guys talked to me. A couple of people had expressed interest in doing something on social networking and how that ties in with medicine. That's what Genomera is all about, and I'm happy to talk to anybody about it. I want to actually skip this part so we have more time for questions. So pardon me as I go through this. But basically, we're running crowdsourced clinical trials at Genomera. The idea is people can try simple things like vitamins. Do they work? Do they not? Let's pool our data. Then I get my information about whether this worked for me. And if I put it out there, we can all see whether it worked for us and see if there are patterns genetically or otherwise. So, to finish up on two things, I think there are a couple of pieces of advice I want to leave you with. One is the idea of not trying to drive exponential trends, but finding the existing exponential trends and hitchhiking. Don't be the guy who's building the sequencer. Be the guy who is using the stuff coming off of the sequencer in ever-increasing amounts. One of the things I think that is important, and here is an audience full of interdisciplinary thinkers. Most of you guys are here because you have got two good careers behind you in one way or another, and you have found ways to work in that overlap. Find those interdisciplinary thinkers, encourage them, find different ways to do it yourself. I won't go through my big pieces of advice here, except to say interdisciplinary work is some of the hardest work, but it's the work that's required by all the grand challenges of the 21st century because they all exist where these pieces overlap. And I think, and I mean this with love, but being at Singularity University, take some of the things we say with a grain of salt. Be skeptical and inspired at the same time. Even brilliant minds with flying cars, I think we're going to get there. It's just didn't happen in 1954, right? Don't let a fancy presentation make you bet the bank on something unless you really understand the fundamentals. And find people who understand the fundamentals. who are actually doing things. Look for people who are working high up on the evidence ladder whenever you're looking in medicine. And I think the most important thing is keep asking questions. That's how you get to the bottom of these pieces. The smartest and best people that I work with in any of these fields are the people who walk into a room and instead of sitting and nodding, they'll say, "Excuse me," and none of you guys have done that, which you're a sleepy bunch or not very interactive, and that's okay. But excuse me, but I don't believe that. How does that work? Or excuse me, you really glossed over this whole DNA structure thing. Let's talk about that a little bit more so I can understand the rest of your presentation. Do that, and do that here, because every one of us who is up here is here less because we get to gab and more because we are really wanting to hear your questions. My very favorite thing about coming here is to hear the things that you're concerned about in your questions. And some of my best collaborations have come out of this space and this audience. The other thing I want to do is there's sort of a set of slides that we're trying to work into each presentation. This is my idiosyncratic version of that. This is not the necessarily Singularity approved way of doing it, but I don't do a whole lot of background. Like I don't say, do this background reading, do all these things, because you guys are busy. How many people are going to really get to this? Nobody's going to get their bachelor's in molecular bio so they can be up to speed and listen to this presentation. However, if you've listened to anything in here and you want to know a little bit more about the background on molecular bio, I love this cartoon guide. And it's a book, it's a cartoon book, which don't let that make you think it's stupid or for kids. It's a fantastic book, and so it's a quick read. It's got good pictures, and it explains basic concepts. Misha Angrist was one of the first human beings to get a full DNA sequence. And he took his DNA sequence, and he took his full medical record, including every medicine he's ever taken for depression and his depressive symptoms, and put them out on the web as part of the Personal Genome Project and said, here is how I think the future happens for medicine. Here's how I think people are going to have to share things for us to all understand what this means. This is a great book. The $1,000 Genome is sort of an industry-wide look at cheap DNA sequencing and who are the players and the drivers there. Biopunk is about this kind of garage biology movement. And I keep threatening that I'm going to write a book, and I keep saying, you know, it just takes too much time. And then I look at, I said that in front of Peter Diamandis once. I was like, I just don't have time to write a book. And he looked at me, he goes, man, I'm writing a book. I was like, okay. If Peter can do it, I'm going to do it. So I've got something that is going to come out probably in the next year. If you really want some background, if you really want to sit down and get a napkin out and sketch and say, this is what PCR is and how it works, or whenever I say SNP, this is what I mean, and yeah, whenever your grandmother got this disease, I think that it's probably this. Let's open up the internet. We've been talking about doing sort of an official office hours, like some hangout time at night, and it's not up officially yet, but what do you guys think? Is anybody interested in that? Cool. Okay, so I will come Tuesday, maybe Wednesday night, but Tuesday night for sure after David's talk. Wednesday, I'm going to try to be here late, and man, I will hang until the last person leaves or my eyes close. And there is no such thing as a dumb question, because if I can get you to understand something you didn't understand whenever you walked in this week, I have done what I wanted to do. So look for me and let me know, and let's get together. Convergence, I just want to mention a lot of the stuff that we're talking about overlaps with what Daniel had mentioned yesterday. The idea of nanotechnology, especially wet nanotechnology, is going to overlap with at least one of the presentations on Friday morning. David Rose is doing this entrepreneurship thing on Tuesday. And of course, in the biotech track, I tend to cover the reading part, the DNA sequencing part. And that's easy because we're going through exponential growth and it's fun to talk about the numbers. The really cool guy is coming up next. Andrew Hessel is going to talk about what happens once you have all of these letters in a row and you can start writing and changing things. And just to breeze through this, so take-home messages, DNA sequencing is getting exponentially cheaper, and if it can drop this much in 15 years, 2020, I will bet anybody the price of a human genome sequence in 2020 that it's going to be a penny. And if it's not a penny, I'll pay for it. So. The other big trend is the democratization of biotechnology. By getting more tools in more hands, I think you got not just breakthroughs, I think you get a renaissance. Resources, people always say, hey, how do you keep up with this? What's the best way for news? What is your favorite, used to be your favorite magazine, your favorite journal, then it was your favorite website. Man, I now believe more in Twitter. And this is such a hard thing for me to say because for me, Twitter hate was the new black, you know? I just didn't want anything to do, 140 characters, right. But I now decide which scientific journal articles I'm going to read by the scientists who have read them and posted them, posting their comments. So I like Twitter and I like the things that go on there. The thing is you got to pick a different group of people about every other week to watch. There are some groups that have websites that I think are particularly up on current news pieces, and look at the blog at Genomes Unzipped. If you were a legal or regulatory person, the Genomics Law Report, and we've had people from both of these organizations come and be temporary faculty here. The Quantified Self movement, and then for the Garage Bio, DIY Bio, and Watch Bio Curious. Some interesting things are going to happen there. And then finally, companies to watch. So, and I can leave this up and I can talk about these, and these are some guys we can talk about later on, but let me just kind of point out under informatics. So I think informatics is a big deal. There are 2 companies there that are really stealth, actually 3, StationX, Personalis, and NewMedi. And all of them are doing things with computers that will really determine what happens with the medicine piece. And for the next generation sequencing, the guys who are going to knock Illumina off of the block, I'm kind of betting on Oxford Nanopore, although we've got some hometown boys, Halcyon Molecular, who think that they can do them even about 100 times better. And diagnostics, if you haven't heard the story of Genomic Health, talk to me about that. Breast cancer diagnostic that is one of these 10-year-long overnight success stories. But probably the real next big thing is going to come from a garage near you. So that's what I think. I really appreciate you listening to me, and I hope you will look at this future where we can ask more questions and be happy with it. And I hope that we have some time to ask some questions. Thank you.
You spoke a little bit about the anti-aging study with the super centenarians, and looking for the differences in their sequence versus a normal person, and perhaps doing gene therapy to make normal people more like super centenarians. If you're trying to tackle anti-aging, I understand that approach, but I've also read a bit about this telomerase. I can't say I fully understand it, but my just back of the envelope understanding is that it's something in each cell that starts off long when the cell is brand new, and a little piece comes off as it ages, but that people have been able to put more telomerase in a cell and make it basically reset it back to 0. So tell me, what is the status of anti-aging, and if you were going to put research into anti-aging or actually reversing aging, would you go the path number 1 with the gene therapy, looking what makes people grow super old, or is it kind of a telomerase-based research project?
Fantastic question. So in a capsule, how do we not get old? And I guess we're kind of all working on that, right? The first part, I was going to say, I think whenever you do some of this longevity genetics research, you're not just limited to, hey, we found the good genes, let's go ahead and do some sort of genetic therapy or genetic engineering to change those good genes. We can actually take sort of a holistic approach where we say, hey, we found these good genes are in these pathways. Here in your pathways, those genes are under or overstimulated. What can we do with lifestyle drugs or something else to sort of shift your pathway flow. And I don't mean to sound hippy-dippy holistic, but like quantitatively, so that these proteins are getting the right workout, these metabolites are at the right levels. Ray Kurzweil has done a fantastic job with Terry Grossman in at least 2 different books where they talk about some of the ways to do that before we have a mature genetic engineering. But the telomerase piece is interesting. I'm not personally a huge believer in the telomerase therapies, but let me do a real quick thing. How many people have heard about the telomeres? So, cool. So the super basic piece is your genes are these long pieces of, your chromosomes are these long pieces of DNA that have your genes on them. On the ends, you've got these pieces that are kind of like the caps on shoelaces, and basically every time you make a copy, they unravel a little bit. Most cells are not equipped, most tissues in your body, with anything to repair that, because it's this natural limit. You need to go like, 40 generations of cells or whatever, it's called the Hayflick limit, and then they just naturally die, and that's what happens. By adding telomerase in, or doing some other kinds of engineering, you can encourage this to be repaired, so this is not a cause of cell death and cell senescence. One of the issues there that people who have really gotten hardcore about it, they found that yes, you can do that, but also whenever you start to do that, you knock out one of the natural controls to cell growth, and you see some things that start to look a lot like unrestrained stem cell activity or cancer. So, and I'm not saying that that keeps this from working, but it's this added problem where you've got to throw some things at it. There is now, I think, 2 different companies, and if anybody knows the names, I think one is Telome Health, and there's another one where basically you can send in a skin sample, a spit sample. They will measure your telomeres and then suggest things that you can do to, address any problem. See if you are out of spec, if your telomeres are especially short and you maybe need to do something, or if they're especially long, then you can, invest in long-term investments. But I think we're still at the early stages. Part of it is longevity. Old age is not seen as a disease. Cardiovascular stuff is seen as a disease. So we have this whole institutional problem of how do we treat something systematically whenever you've got, and the funding institutions and the health institutions don't have a place for it. That's starting to change, though, and I think we're all concerned about it. Aubrey de Grey, who is one of the advisors for Singularity U and sometimes shows up, his books are great to read about sort of this systematic treatment of old age. So, but we can talk more.
I have the microphone here. Right, I think you said that there are about 1,500 FDA-approved drugs, and about 20 of those have some kind of correlation established to the genetic information and how the individual interacts with that drug. So I'm wondering what's involved in getting that information for the rest of the 1,500, and who's paying for that?
It's kind of interesting. So yeah, 1,500 drugs approved. Some of those are approved because they're like aspirin. If you took aspirin, you said, I discovered aspirin today. It's 2012, it's the wonder drug. FDA would probably never approve it because there's so many things that aspirin does. But out of those 1,500, we have about 300 where we have really detailed genetic information. And that's because as they went through that approval process historically, we had started to have the tools available in about the 80s to go ahead and look at what are the target protein molecules and what are the genetics of those molecules. How are your and my proteins that are targeted a little bit different? So really any new drug that comes through there, we're going to get investigations of that. The old drugs, that's the problem. There have been some government-funded ideas or programs to start looking at that. There's a great database at Stanford where I got most of the information on those 300 that we have. And I can share that whenever I remember what it is. I need to take the memory-enhancing drug. But economically, there's no incentive for a company that's got a drug that they sell especially if it's off patent, to say, let's go study this some more and spend money. And if it's on patent, they say, yeah, let's go study this some more so we can tell our 100% population that uses it that it really only works on 20%, which has been this horrible back and forth. Finally, these companies are believing, and you know, I just got out of an interesting talk at the Keck Institute last Friday, where the guy who was in charge of this program for Merck until about a year ago said, yeah, we now finally believe we would rather sell a drug that works really, really well for 20% of people than one that works so-so, and we're going to have better market share. And he said, and here's how we ran the numbers. And everybody's like, oh, thank God. So that's, but it's really kind of one of the big economic problems of our time, I think.
Many thanks for the presentation. It was amazing. Congratulations. Just one question. I would like to ask you if supranational organizations are already leveraging upon all this knowledge to help and use all this, I would say, know-how in order to, I would say, make, I would say, part of the world population that live in certain area more stronger compared to our general disease. For example, who lives in a very warm area of the world, make it more stronger to live there, or whatever. So if it is already in use, and if not, which is your expectation?
And to make sure I understand the question, let me kind of echo it back and shake your head or tell me yes. So basically, looking especially at like disadvantaged populations, non-first world populations, are there programs or what are the things? We've actually, I think, done surprisingly well in that there are a lot of charitable organizations that really address this. And the first one that comes to mind is the Gates Foundation, where they have funded some amazing research in this. So you guys are going to get to visit, and it's a great visit. It's quite a privilege to kind of go in the gates of Amyris later this week. One of the stories of Amyris, they had decided they wanted to figure out how to take a drug that was necessary to treat malaria, fairly expensive to produce and transport, and go ahead and make it so that you could grow it in yeast, which would mean that you could go ahead and distribute this like spores. You could grow this, you could, they talked about growing it in bananas, so that instead of getting a vaccination, you ate a banana, kind of a thing. And these are the kind of projects that are going on there, and that was something that was sponsored by the Gates Foundation. And I kind of expect basically all drug production and all, really at some point, all materials processing and all industry to start looking a lot like a microbrewery. Instead of having a big industrial infrastructure to make a material or to produce some complex drug, you've got some guy brewing stuff in his basement. And you know, that's good and bad. But to get back to how are we using this on sort of, how are we using this with third world populations? One of the problems is, you know, you kind of depend on this infrastructure to do biotechnology. And you need electronics, air conditioning, lights. Most of these are fairly high-tech machines that run in a stable environment. And you depend on the other infrastructure people don't take into account is FedEx. If you can't order a chemical and get it overnight, you can't do an experiment or do a treatment. So there are a few people finding really clever ways to attack both of those problems. The neatest thing I've seen is a PCR machine. You know, we've got one in the lab that's 8 years old that's this big and 600 pounds. You guys saw that open PCR machine that's about this big. There's one that'll fit in the palm of my hand now and is run off of a 9-volt battery. And you can actually stick it in your pocket or a backpack and take it into the Amazon basin and use it to do diagnostics on Chagas disease and things like that. So this democratization, sort of mobilization of equipment and technology, I think is going to help a lot. The other thing, things are getting so cheap so fast, kind of the cast-offs from the first world, I think are going to drive a lot of this. There's somebody here from Brazil that I want to talk to about some of that. There are some interesting things. I'd recently taken a trip and I'd seen some comments on a comment form, but I'm going to stop talking and take more questions.
I would like to know regarding if something is being done to map a room or an environment to see the living creatures in this room, since to look for contamination or to map the biodiversity.
That's a fantastic question. The one section I didn't squeeze into my talk last night was on this idea of metagenomics. So instead of just taking a sample from an individual and sequencing them, or even from a whole population of a species so that you know what the variation is, the idea is, what if you take a gallon of seawater and find every organism in there and sequence the DNA that they've got, or a scoop of dirt on the ground, or something from the sewer, or something like this. Metagenomics is the name for that. And it's really become this almost weirdly, fantastically successful thing. People said, well, that's impossible. You can't do that in distinguished organisms. People are now finding ways to barcode, in effect, genes, figure out where they came from, and start to map biodiversity. One of the huge, interesting findings from this is, we thought that we knew something about 10% of the species on the planet. That percentage is shrinking as we see how many more different species that fill these niches and are so much more diverse than we had suspected are. The other thing that is interesting to me is, kind of a hacker, is this is a great part source for doing something in biotechnology. In the past, whenever pharma wanted to find a new drug, one of the things they do, they'd send people all over the world to take soil samples, because soil is teeming with bacteria that fight wars with each other. So you can find good antibiotics and things like that. And now here's our chance to find proteins from all over that we can use to manufacture other things, whether they're drugs or materials. Craig Venter, who was the guy who did the non-government funded Human Genome Project, whenever he sort of retired and sailed around the world, he took a big bucket with him, basically, and would bring up seawater and did this really elegant program of sequencing the microbes that they found in the seawater. And they found, I think, 1.5 million new species that had never been recorded, that sort of thing. So we're just starting to scratch the surface.
Amazing presentation. I'm still blown away. So I can't help, listening to this and drawing incredible parallels to the personal computer revolution in almost every way. And it seems like where, semiconductors and hardware were really the, first big things that enabled everything to come down to grasp for somebody like me, or somebody in my position. It seems like we're getting just there with biology right now, whereas personal computers was humanities and technology, this is biology and technology. So I'm just wondering, so right now it seems like if I was to draw that line and where we are, it's when the first clunky hardware came out and the first graphical user interface like 23andMe will kind of make it easy to understand, but it's still not even close to anything sophisticated. So one question is, is it going to be 30 years before we see the biology iPad equivalent, or how long is it going to take? And where do you kind of see that going? Because this is the first industry after personal computing that really requires new hardware that's never been seen before. So where do you see that going? Another question is, what countries, if any, outside the US are kind of making strides in bringing biology and technology together?
Great questions, and remind me if I start to leave off countries, because I want to hit that. So, I love the bio-curious concept, the idea that, hey, let's just take all this high tech that has been in the ivory tower, plop it down in a neighborhood, invite in teachers, school children, people who want to start companies, everybody, start playing with it and see what you can do next. And we've had exactly that kind of diversity of people doing things. We have very consciously, copied the, if you will, the DNA of the Homebrew Computing Club, which was literally you could throw a rock and hit it from across the freeway. We said, hey, let's go ahead and set this up just like those guys did. Let's make sure it's an open atmosphere, people can share things. We'll put as few restrictions, but we'll keep people from hurting themselves or others. So I really think we are right at that stage where those guys kind of opened their doors and said, here, we're the Homebrew Computing Club, let's start having meetups, and who did something neat this week. I think in a lot of ways, 23andMe, and people kind of get up and down on them both. They're the pioneers, so they've got the arrows in their back. They are sort of our DOS, you know. They said, here you go, and here's some things, and isn't it great to have the command line, and you only have to have these 4,200 commands to see what's going on, which is a lot better than what you were doing before, but you can see that it should get a lot better. The visualization of these and bringing it to the grandma level, and that's everything I work with in this space. We do a grandma test, and I don't mean that pejoratively, like my grandmother is brilliant. She's just not a geneticist. And if I can't explain something to her, then I'm not hitting it right. And it's funny because, you know, we did her 23andMe, and whenever I talked to her, she would say, hey, you know, I saw there's a new research article out, and I was trying to gauge what this is, and it's only got 3 stars, but since they had a high control size in their cohort, and I'm just like, Grandma, where did you learn this? And she's like, I read the paper, what do you think? So I think we are at early days, and I think we're kind of just starting to even climb the hype cycle. There's going to be, again, for every bit of this industry, these places where people say, oh, there was this promise and it wasn't realized. Man, it's coming. Amazing things are happening, whether they reach individual people in the populace and individual societies, kind of the non-first world societies, as quickly as they can. In some ways, I think some of this is going to work even better outside of first world societies, because just like if you are going into Africa, you don't start stringing up telephone poles, you start putting in cell towers, right? You do this different infrastructure, and it has this amazing transformative effect. People are going to economies that depend on different profiles of workers, economies that depend on different kinds of populations are going to need genetic diagnosis and testing more maybe than the US. People with different regulatory profiles, and we're already starting to see that. India is a great hotbed for developing some new diagnostics because it's got a large population. Man, the bioinformatics people there are in a lot of ways so much better. There are about 10 times as many of them there as there are in the US. And English speaking, so they all read the same journals, and they read them and they come up with these great computer programs. They're like, why aren't you guys getting on the program? They talk to us in the US and say that. China is always the big dog in the room. You just don't know what they're going to do next. But as an example, saying, hey, let me get out my checkbook and write you a check for, you know, $64 million, and we're now the premier sequencing center of the world. We do more sequencing than everybody else combined. They can do those kind of things. Brazil and Chile, I think, are doing some really interesting things, especially with environmental pieces and biofuels, and we'll be talking more about that. The regulatory landscape is really complex worldwide. I want to mention Saudi Arabia has a program where they are sequencing, full genome sequence, every one of their citizens. And part of that is a little spooky to me, but it's to identify bloodlines and who is really a citizen and who is not. A surprising number of nations in the world have citizenship based on bloodlines, you know. So,
Have they started that?
yeah, and they're doing that with Life Technologies. Life doesn't like to talk about it because it makes them look a little complicit in something that's maybe ethically yucky. So, but one of the things that's going to be cool is they're going to have this great database, and if they can cross-reference that with their medical database, they're going to have a profile on everybody in the country and be able to do preventive health care like something you would see in the Jetsons. So, and that's not what they're setting out to do, but that's going to be the fallout from it. We're going to worry about some of the privacy and regulatory stuff, and we should. You know, I don't have my kids' genomes out on the web. I've got those locked away. So.
I read somewhere a few months ago that China took 2,000 of the top geniuses and comparing them with 2,000 normal people and break down the entire genome sequence of those to find out
The scariest thing about all of this stuff to me is not, what are we going to do medically, but the things we're going to find out and say, really, this is Gattaca. This is genetically determined. You're going to be able to do this well. This person can do it this well. As much heart as you have, you will never be the long-distance runner who's got the breathing capacity and the fast-twitch muscle tissue to do this genetically, and we'll have that gap in what we can scan and what we can change for a while.
One question. Did you already sequence an astronaut in space to see how DNA is affected? And the second question is, according to the information that you obtain of a sequence of a species, is that enough information that in the future somebody can replicate that species, or you need a biggest amount of information?
As far as I know, there haven't been any full sequencing experiments done in space. Excuse me. There have been some experiments on how methylation, how epigenetics is affected by microgravity. And so far, not much has come out of it. Part of it is DNA, and this kind of speaks to your next question too, is really hardy. It takes a lot to change it, although you see some gene expression changes, and there have been gene expression tests on different astro animals and astronauts. There have been cases where DNA is extracted from woolly mammoths, Neanderthal tissue. This is stuff that's tens of thousands of years old, and it's intact, sometimes by being frozen, sometimes not. And DNA lasts and lasts and lasts for a long time, so everybody's leaving their mark on the world one way or another. We do have enough DNA from some of these fossil animals where theoretically we could put it in a cell of something that is close enough and reboot it and pull a Jurassic Park, or even, like a Neanderthal reboot. But there's some other issues there, some of the least of which are not ethical, right? But I think that's fascinating. Some people talk about, do we want to have, instead of a lot of conservation, just repositories of DNA, which scares me a little bit. I think that's a great backup, but if you do that, don't say, and so let's wipe out this rainforest because we've got it all on disk. I don't think that's a good idea at all. 2 more questions? 2 more minutes, excuse me.
As we move, and I think you said the next guy later is going to talk more about the writing process as opposed to the reading, but I wanted to ask you, while you're here, how quickly, as we start genetically modifying ourselves, do we sort of bifurcate as a species? I mean, you hear about that sometimes on the fringes, but I mean, is that a real concern? Is that not a concern? And what are your sort of thoughts on that?
I think it's a real thing that can and will happen in more of a far-flung future, but really there are enough little bitty edits we could do to eliminate disease, to make sure that you've got the right amount of fast-twitch muscle tissue, you never get a migraine, you are a morning person, and all this stuff, where we would be entirely human, just like any other human. It's just kind of the best bits. Now, there are ethical issues with that, but yeah, it's a while, unless somebody said, I consciously want to produce a non-breeding subspecies before any of that would happen. So, but it's a great question.
We have to end there, unfortunately.
Guys, thank you so much.